carotenoid pigments, they are going to be red, orange, yellow in color, and they would be mostly or found in the chromoplast fully and partially in the chloroplast. Okay. Now, carotenoid pigments can be of two types, carotene and xanthophyll. Now, before we go into the differences between carotene and xanthophyll, do try to understand one very important thing about carotenoids, that what is the main function of carotenoids? The main function of carotenoids would be that they would save the chlorophyll pigments or protect the chlorophyll pigments from photooxidation of light, means from damaging effects of light. When very high light intensity falls onto the chlorophyll pigments, they might undergo damage. So from this damage, which is called photooxidation, who would protect the chlorophyll pigments would be carotenoid pigments. Understood? That's why carotenoid pigments are also known as shield pigments. Okay? Understood this? So, now carotene and xanthophyll, these are the two types of carotenoid pigments. Carotene are going to be, the formula C40H56. They are mostly red to orange in color. Okay. Now, the most abundant carotene pigment would be beta carotene, and which is found in abundance in the carrot. And beta carotene inside the animal bodies will be used to synthesize vitamin A. Okay. On the other hand, xanthophyll pigments, they are oxygen derivatives of carotene. So basically they have the formula C40H56O. Okay. They are mainly yellow in color. And the most abundant xanthophyll pigment, which can be seen in winter foliage, is lutein. Lutein has the formula C40H56O2. Alright, likewise you know about the xanthophyll pigment, that is fucoxanthin, which provides brown color to the brown algae. That would have a formula of C40H56O6. So likewise, there would be various types of xanthophyll pigments known. Alright, understood this? Now, for a very important thing that we are going to discuss is the sunlight, the solar radiation. Let me give you an idea about it. Solar radiation. So, you know very well that sunlight or solar radiation consists of various frequency of radiations, isn't it? The highest frequency would be for, say, cosmic ray, then gamma ray, X-ray, UV, then visible light, then comes infrared, microwave, radio wave, isn't it? Out of the various frequencies of radiations that are present in sunlight, mainly for photosynthesis, we require visible light or visible spectrum of radiation, isn't it? So, visible light allows photosynthesis. Visible light has the wavelength range of 390 to 760 nanometer. Okay? Out of which 400 to 700 nanometer range is the one in which photosynthesis occurs in the most of its efficiency. So that's why this range of radiation is called PAR, photosynthetically active radiation. Okay. So let us finally split up the entire visible light to observe that which of the lights are going to have which type of wavelength and where exactly photosynthesis will occur. this is the entire split up of the visible spectrum of light, and we can divide it into 7 colors. And out of all these colors, you know that photosynthesis mostly will take place in this section, 470 to 500 nanometer, that is blue radiation, and 650 to 760 nanometer, that is red radiation. So do remember this much. Blue light 470 to 500 nanometer, whereas red light 650 to 760. Remember this range. Okay. Let us first start with absorption spectrum. Now see, absorption spectrum means the amount of absorption done by a photosynthetic pigment when various wavelengths of light are going to be exposed to it. Okay, it is basically a curve that is depicted on a graph paper. Okay, so suppose this is the graph. Here we write about absorption, and this is the wavelength. Fine. 300, 400, 500, likewise. Here 5, 10, 15, 20, the amount of absorption that can happen, likewise absorption. Alright. Now whenever you take a photosynthetic pigment and you try to measure that particular photosynthetic pigment is absorbing which particular wavelength of light, or how much of energy of a wavelength is it absorbing, that particular measurement can be done. Usually it is done by using a method or using a instrument that's called spectrophotometer. Okay, spectrophotometer is used to see the absorption of light. Fine. So whichever photosynthetic pigment you try to find out its absorption spectrum, you will find that the absorption spectrum graph will show you 2 peaks. One of the peak will be from 470 to 500, that is blue light range, and another peak you will observe in the red light, that is 650 to 760. This range you will find another peak. Okay, so that means any type of photosynthetic pigment you take into consideration, it will always be absorbing mostly blue light and red light. Okay, understood this? Now for action spectrum, in action spectrum, this is also nothing but a curve plotted on a graph paper, but this time we are going to see the rate of photosynthesis. In which particular wavelength of light, rate of photosynthesis would be maximum. Okay, so again the same graph. Alright, again you will find that for action spectrum as well, you will have 2 peaks like this. One peak will be in the blue light zone, another will be in the red light zone. So by this we can prove again that photosynthesis does occur in visible spectrum of light, but then in the visible spectrum it is blue and red radiation which favors photosynthesis the most. Okay. Now, if we compare the absorption of chlorophyll A, B and carotenoids at different wavelengths, we will find a graph like this. This is suppose for chlorophyll A. Okay. This is for chlorophyll B. and this is for carotenoids. Means chlorophyll A will be absorbing blue and red light, which will be happening almost in the same pace, but of course red light will be absorbed a little more than blue light. In case of chlorophyll B, chlorophyll, that is the blue light will be absorbed much more in comparison to red, but in case of carotenoid pigments it will be absorbing mostly green and blue light. It will not be absorbing at all the red light. Understood this? Clear? Bluish green zone is the zone where carotenoid pigments will be absorbing the most. Rest of the places, rest of the wavelengths, it will not be absorbing. Alright, so this is the comparative study of chlorophyll A, B and carotenoids, how much absorption it does in different wavelengths of visible light. So I hope all of you are clear about action spectrum, absorption spectrum. So now see, photosynthetic units are a small group of photosynthetic pigments which will together participate in the light reaction. Okay. Now photosynthetic units are going to be having a distinct structure like this. The outermost layer is known as the antenna complex. Then comes core complex, and finally remains the photocenter, or we can also call it reaction center. Now what is the meaning of these things now? Antenna complex is mainly carotenoid pigments. Core complex will be a mixture of chlorophyll A and B, but reaction center is a single chlorophyll A molecule. Okay. Together the core and the antenna molecules forms the light harvesting complex. Okay. So what actually will happen, you see, in the photosynthetic unit, when sunlight will fall on the photosynthetic unit, at first the antenna complex will absorb the light, and by resonance it will get excited and it will pass on the energy to the core complex. Similarly, the core complex will also get excited and will pass on the energy by resonance to the reaction center. Finally, the reaction center, on receiving so much of energy, will get excited and it will be the one that will participate in the process of light reaction. So it is this reaction center chlorophyll A that will actually participate in the light reaction. Rest of the pigments, they will simply be allowing all the energy to be transferred to the reaction center so that it gets excited. Understood this? So reaction center chlorophyll A will finally be actually participating in the process of light reaction. Do remember this. Now, the photosynthetic units can be of two types. PS1, photosystem 1, and PS2, photosystem 2. They have been termed according to the sequence of their discoveries. Now, PS1, it is mainly found in non-appressed thylakoids and stromal lamellae. First thing, alright, it consists of photocenter or reaction center, which is photocenter or reaction center, that will be consisting of a chlorophyll, or let me write it here, photocenter that will be consisting of chlorophyll A 700. That means it will get excited in 700 nanometer of radiation. Apart from this, it will also possess light harvesting complex 1 and certain electron carriers. Alright. On the other hand, for PS2, it will be mainly found in appressed thylakoids, and it will consist of photocenter or reaction center which will have a chlorophyll A 680, light harvesting complex 2 and certain electron carriers. Apart from that, oxygen evolving complex. Alright. Now, what are the electron carriers associated with PS1? It is mainly A1, that is a spatial chlorophyll molecule. Then a quinone. Then there will be iron sulfur proteins, ferridoxin, cytochrome B6F complex and plastocyanin. Alright. Whereas for PS2, the electron carriers which will be present will be pheophytin, plastoquinone, cytochrome B6F complex, which is common to both PS1 and PS2 then, and plastocyanin. Alright, these are the components of PS1 and PS2 which you need to remember very prominently so that you can understand the light reaction very properly. Because in the light reaction you need to first know what are the components of PS1 and what are the components of PS2. Okay, try to mark it very properly and try to memorize it before we discuss light reaction. Finally, we have reached the mechanism of light reaction or photophosphorylation. Okay. Light reaction is also known as Hill's reaction. We can also call it photochemical phase. Okay. So light reaction can be basically, here we are going to discuss two types of mechanisms: non-cyclic photophosphorylation and cyclic photophosphorylation. Non-cyclic photophosphorylation is also known as the Z scheme because of the zigzag pattern in which it would occur. Okay. Now see, non-cyclic photophosphorylation takes into account both PS1 and PS2. It starts with PS2, finally it would end in PS1. Okay. So let's see what would be happening in non-cyclic photophosphorylation. See, at first when sunlight will fall in PS2, P680 is PS2, isn't it? So when sunlight falls on the PS2, the reaction center chlorophyll A will get excited and it will liberate 2 electrons. These electrons will be taken up by the early electron acceptor pheophytin, and pheophytin will transfer the electrons to plastoquinone. PQ is plastoquinone. Plastoquinone, as soon as it will take the electrons, it will take up 2 protons from the stroma and will form plastohydroquinone. As the electrons are further passed on to the next electron carrier, the proton will be now shifted to the lumen of the thylakoid. In between the 2 membranes of the thylakoid, the space that remains is called the lumen. Okay, there the protons will be accumulated. Now the electrons will be now taken up by cytochrome B6F complex, and from there to plastocyanin, and from plastocyanin the 2 electrons will move on to PS1, that is P700. Now here you need to understand something. See, when PS2 got excited, it liberated 2 electrons, but to bring it back to the ground state, we also need to provide it with the 2 additional electrons that it has lost. So for that, here the oxygen evolving complex will conduct the step of water splitting or photolysis of water. In photolysis of water, the water will be split into 2 electrons, 2 protons and half of oxygen. These 2 electrons will be transferred to PS2 so that it gets back to the ground state again, and it can again be starting another photoreaction. Okay, it will be like it can start another round of excitation. Alright. The 2 protons, it will simply be getting accumulated to the lumen of the thylakoid, and the oxygen, being the byproduct, will be released outside. Okay, so this is the step in which oxygen will be released during photosynthesis, and the oxygen is of course coming from water, not from carbon dioxide. Do remember oxygen evolving complex. This is an enzyme complex that will require certain mineral elements for it to function, and they are manganese, chlorine and calcium. Alright. Now, by the time also, see, PS1 and PS2 will get excited at the same time, isn't it? So, by the time the PS1 is excited and it releases 2 electrons, it will also have to get back to the ground state, isn't it? To get back to the ground state, the 2 electrons that it would require are coming from plastocyanin. So these 2 electrons from plastocyanin being transferred to PS1 is actually going to bring it back to the ground state. Okay. So now let's see what will happen to the 2 electrons which are liberated from PS1. So these 2 electrons will be first accepted by a special chlorophyll molecule A0, then it will move on to a quinone A1, and finally the electrons will pass on to iron sulfur protein and then to ferredoxin. From ferredoxin, the 2 electrons finally will be passed on to NADP+. Now, the enzyme that will be, converting NADP+ into NADPH+, it is the NADP reductase. Now, the enzyme will be present towards the, stroma, towards the outer side of the thylakoid membrane. Hence, it will take up 2 protons from the stroma to finally convert the NADP+ into NADPH+, H+. Alright, so this is the entire Z scheme, at the end of which you are seeing NADPH+, H+ has been formed. But then how is ATP going to be formed? By the Z scheme or by the light reaction. This being the mechanism of light reaction, I haven't mentioned yet how ATP will be formed, isn't it? ATP plus NADPH, both are the product of light reaction. You are directly seeing how NADPH is being formed at the end of the process, but there is no mention yet of ATP, right? So let's see how ATP will be formed, and this formation of ATP will be explained by, or it will be explained under chemiosmotic hypothesis that was proposed by Peter Mitchell. Okay, so chemiosmotic hypothesis. See, in the Z scheme, there are 3 such steps which will do a very important thing, that is, suppose this is the thylakoid membrane, this is the lumen, and this is the outside, that is stroma, right? Due to 3 crucial steps in the Z scheme, proton concentration in the lumen will be high, proton will be high here, okay, and proton will be low here. That means a proton gradient will be set up. Now, which are those 3 steps? One of the steps is this, photolysis of water. Here, 2 protons are directly being accumulated in the lumen, right? Next, second step is this, where you see that as soon as the 2 electrons are passed on to plastoquinone, 2 protons from the stroma are being taken and are transferred to the lumen, right? So proton in the stroma is reducing and in the lumen it is increasing, right? So this is one step. Finally, this is the last step. Here, when NADP+ is getting converted to NADPH+, H+, at that time 2 protons are being taken from the stroma, right? So due to these 3 steps, one is this, then this and this. Finally, what you are getting to see is that proton concentration in the lumen is increasing, whereas in the stroma it is decreasing. So just like any substance in nature would always want to move from higher to lower concentration until an equilibrium is reached, isn't it? So same in case of proton also, the proton would try to move from lumen towards stroma, right? But then in between comes the thylakoid membrane, which is impermeable to proton. So simple diffusion of proton is not possible. The only thing in the thylakoid membrane that will allow proton to be transferred to the stroma, which is going to be proton permeable, would be F0 F1 subunit, or we can call it the ATP synthesis. Okay. So ATP synthesis or F0 F1 subunits, these are going to be similar to mitochondria. Okay. So they are going to allow the proton to finally transfer from the lumen towards stroma. So every time 3 protons are pumped through the ATP synthesis, as it is passed from F0 unit to the F1 subunit, there will be certain conformational changes that would take place, due to which one ADP plus inorganic phosphate will combine and form a ATP. So every time 3 protons will be pumped through the ATP synthesis, one ADP will be converted to one ATP. Is that clear? So by the help of the Z scheme and these 3 crucial steps, we are finally getting a proton gradient, and due to the proton gradient, when the protons are being pumped from the lumen towards the stroma, at last we will have ADP being converted to ATP. That's how we will be producing ATP during the light reaction. This whole thing is explained by Peter Mitchell under chemiosmotic hypothesis. Understood? So this is how NADPH as well as ATP will be formed by non-cyclic photophosphorylation. I hope all of you are clear. Now, the next thing we will discuss is cyclic photophosphorylation. Now, finally, for cyclic photophosphorylation would occur in low light intensity or when carbon dioxide fixation will be inhibited. So when carbon dioxide fixation is not going on, the ATP and NADPH formation is not required, isn't it? So at that time, cyclic photophosphorylation would occur. Do remember, cyclic photophosphorylation involves only PS1, okay, and it does not involve PS2. Hence, photolysis of water or splitting of water step is going to be absent here, okay. So as you can see here, as sunlight falls onto PS1, it will get excited and liberate 2 electrons, which will as usual be transferred to spatial chlorophyll molecule, then to A1, and finally as usual that you have seen in the non-cyclic photophosphorylation, it will be passed on to iron sulfur protein, ferredoxin, and finally from ferredoxin, you know that now in the Z scheme, the electron will be transferred to NADP+, isn't it? But then we do not require NADPH formation here. That's why the electron instead will be passed on to Plants-2 which will be taking 2 protons from the stroma as usual and will be transferring it to the lumen, okay, while the electron moves to the next electron carrier. So it will be happening through cytochrome B6F complex, and then from there the electrons will be finally passed on to plastocyanin, from where the electron which was lost will be compensated to PS1 via plastocyanin. Plastocyanin will give the 2 electrons back to PS1. So that's why it is forming a cycle. The electrons are being liberated from PS1 and it is getting back to PS1 again, forming a cycle. That's why we call it cyclic photophosphorylation. Do remember, in cyclic photophosphorylation as well, in this step of a proton gradient is being formed that will give rise to ATP formation. Okay, is that clear? So ATP formation would occur in cyclic photophosphorylation, but NADPH formation will be completely absent. Fine. Understood this? So this is what is cyclic photophosphorylation all about. With that, light reactions would be completed. In the next class, we'll start with dark reaction.